Hi, everyone. We are the Mighty Mitochondria. And today we will be discussing what is the difference between non-Mendelian inheritance and extensions from Mendelian ratio.
When talking about extensions to Mendel's laws, also referred to as extensions, deviations from Mendelian ratios, we refer to the genetic process that differ in phenotypic expressions like codominance or incomplete dominance. These exceptions still follow Mendel's laws of segregation and independent assortment, but the phenotypic ratios differ from what is expected from a complete dominance relationship.
So as we know, a completely dominant relationship is seen here, where we have a homozygote dominant, a homozygote recessive, and heterozygotes. So what we have here in the genotypic expression is a 1 to 2 to 1 ratio, and our phenotypes are 3 to 1, 3 being following the dominant genotype, and 1 being the recessive. The main point of this is to show that when we have an incomplete dominance relationship, our genotype stays the same, 1 to 2 to 1. We still have 1 homozygote dominant, 1 homozygous recessive, and 2 heterozygotes, but our phenotypes are now 1 to 2 to 1, as seen by the blending of the red and white to make pink. Similarly, with codominance, you have 2 different phenotypes and a blending of the 2. So we have 1 spotted, 1 striped, and we have a mix of the 2 as the heterozygotes as dotted and striped.
Non-Mendelian ratios. Extension from Mendelian ratios is not the same as non-Mendelian inheritance, because non-Mendelian inheritance is the genetic inheritance that do not follow Mendel's law of segregation or independent assortment. These inheritances refer to a few cases which include epigenetics, maternal contribution, and a few cases of genome from organelles like the mitochondria or chloroplasts that also do not follow Mendel's law.
In this image, we are depicting epigenetics. And in this image, we are depicting genomic imprinting mechanisms where the methylation of an insulator located between the enhancer and promoter controls maternal imprinting of IGF-2. In this image, we are depicting mice who have exposure to environmental stimulus, which is an example of intergenerational versus transgenerational epigenetic inheritance. Thank you.